Hello, my name is Ravi Vij. I'm from MyCancerHaven, and today in the Conversation with Cancer Expert series, we have Dr. Geoffrey Shouse, who is Assistant Professor in the Division of Lymphoma at the Department of Hematology and Hematopoietic Stem Cell Transplantation at the City of Hope in Duarte, California. And we're going to talk about a drug that has only recently come to the fore in the treatment of heme malignancies, Pirtobrutinib. So welcome, Geoffrey.
Thank you.
So, Geoffrey, just for the audience, can you give us a little primer on the role of BTK in hematological malignancies? And then we can obviously talk about where Pirtobrutinib fits in.
Sure. So maybe starting from the top, we can kind of, looking at the diagram, see that the B cell receptor is kind of the primary start of the signaling cascade. And so we're talking about B cell malignancies here. And in B cell lymphomas, we have CLL, MCL, marginal zone, Waldenstrom's, among many others. But the importance of the B cell receptor is that it can drive growth signals and proliferation signals. And if those growth signals remain on and active, then you can get cancer. And one of the downstream molecules is the Bruton's tyrosine kinase. And there are situations where this molecule or upstream signaling pathways can become active and promote growth of these various lymphomas. And so when it was identified how important this particular molecule was to the growth of many of these B cell malignancies, it became a very attractive target for inhibition. And since then, we've identified multiple different inhibitors of BTK that have been very effective therapies that have kind of revolutionized the treatment of many of these cancers, not just because they work so well, but because they can also be given orally. Patients usually tolerate them pretty well, and their toxicity profiles are pretty well managed.
So you do mention that we've had now got a number of these BTK inhibitors. So just to put in perspective before we launch into Pirtobrutinib, what has come before it?
Definitely. So the first generation covalent BTK inhibitor was ibrutinib. Very effective, does have some pretty significant toxicity, including bleeding toxicity, as well as cardiac arrhythmias, including atrial fibrillation, atrial flutter, and now in some of the more recent studies, even ventricular arrhythmias. Then second generation covalent BTK inhibitors have come out, which include acalabrutinib and zanubrutinib. And they're highly active in these B-cell malignancies, but they appear to have less toxicity and are better tolerated. And now, what's been seen is that there are resistance mechanisms that pop up that allow the cancer cells to eventually kind of overcome the BTK inhibition, and the cancer cells can grow again. And some of these resistance mechanisms are mutations that arise in the BTK gene itself, promoting resistance to these drugs. And so one way around that is with these novel non-covalent BTK inhibitors, and that's where Pirtobrutinib comes in. So it's a next generation selective non-covalent BTK inhibitor. And it's able to overcome some of these other mutations that occur in the binding pocket for the other BTKs.
So it's if I get you correct, where if you truly progress on ibrutinib or acalabrutinib or zanubrutinib, switching between those may not be likely to produce a response. But Pirtobrutinib is potentially going to work even when you've progressed on one of those drugs. Is that what we're trying to say here?
Yeah, that's accurate.
So next, we'll just review the data. So Pirtobrutinib is now approved in a couple of disease states. So we'll start out with the first approval, and I think that was in mantle cell lymphoma. And can you just briefly tell us about this trial and walk through a few slides here and try to educate us about this drug in mantle cell?
Absolutely. So the data for Pirtobrutinib that led to its approval by the FDA in mantle cell was based on the Bruin study. And what we know is that patients who have mantle cell lymphoma but progress after BTK inhibitors tend to do very poorly. And so there's a need for treatment of these patients that works in that setting. And so this was a dose escalation and expansion phase 1 and 2 study to find at first the best dose for Pirtobrutinib, and then to see how effective it was once they figured out that best dose. And it's for adult patients with mantle cell lymphoma who've had 2 or more prior therapies, including a covalent BTK inhibitor in the primary analysis subset. And so they looked at safety, PK, overall response rate, but the primary endpoint for the phase 2 study was overall response.
So what were the results in this population of potentially heavily treated patients?
So they seem to be quite promising, to be honest. So they subdivided the patients based on whether they were exposed to prior covalent BTK inhibitors or whether they were covalent BTK naive. The majority of patients, 90, had seen prior covalent BTK inhibitors, and in this subset of patients, the overall response rate was still almost 60%, which is excellent in the setting of heavily pretreated patients, with a CR rate approaching almost 20%. And for patients who were BTK inhibitor naive, it was almost double that. So the CR rate was over 40%. And the overall response rate was 85, which is excellent, even though they hadn't seen BTK, covalent BTKs before. But overall, very promising. And the duration of response and progression-free survival were also excellent in patients, both who had not seen BTK inhibitor before, as well as those who had. So the median duration of response was 17 months in patients who had become resistant to covalent BTKs, and the progression-free survival was 7 months.
And how well is this tolerated?
For the most part, it seems to be tolerated quite well. The concerns with any new drug is that it might be more toxic than something that was available before, but that doesn't seem to be the case here. So looking at any grade toxicities, especially those that were deemed related to the drug, majority was fatigue, diarrhea, a little bit of dyspnea, some bruising, but all this was mainly grade 1 or 2. The most severe grade 3 and higher toxicities were fatigue, which was only 2.4% of patients, dyspnea, and then 0.6% of patients had hemorrhage or hematoma. And bleeding is always a risk in patients who receive BTK inhibitors, which we've seen with the prior drugs.
So before we move on to its approval in CLL, in mantle cell lymphoma, we have a number of options, including recently approved CAR T cells as well. So where do you see this fitting into the treatment paradigm in your algorithm? If you could just briefly summarize how you would fit Pirtobrutinib into the treatment.
Definitely. I think I would use this for sure in a patient who I thought who had progressed after a covalent BTK inhibitor, especially if I didn't think they were a candidate for CAR T, or if I wanted to try to debulk their disease to try to limit their toxicity when they underwent CAR T. Because one of the main issues that complicates use of CAR T therapy is the toxicity that's seen with currently FDA-approved products. There's a lot of CRS and ICANS that can happen. And if you can debulk the patients beforehand without affecting the T cell function, then it's a good option in that setting. And definitely anyone who I thought was CAR T ineligible, I would probably use this for too.
And moving on to Pirtobrutinib in chronic lymphocytic leukemia, could you educate us about the trial that led to its approval for this disease state?
Yeah, definitely. So the Bruin study was using Pirtobrutinib for previously treated CLL patients. It was a multi-cohort phase 1/2 study. They initially identified what the optimal dosing was for the phase 2 and then expanded. An interesting thing about this study is they use several different groups of patients. So patients who were BTK inhibitor naive, patients who had seen prior BTK inhibitor, the covalent BTK inhibitor, and either had been exposed to prior BCL2 inhibitor or were BCL2 inhibitor naive. And as far as molecular targeted therapies go for CLL, BTK inhibitors, the covalent BTK inhibitors and BCL2 inhibitors are really the go-tos for frontline and early relapse setting. So they're looking at how this drug works in probably the most important patient classes, I would say. Primary endpoints
Sorry.
were mean tolerated dose. No, that's okay. Mean tolerated dose and then overall response rate.
So, and what was the demographics of the population accrued to this study?
So there was a wide range in age, which we can see for CLL, but predominantly the median was older adults, which I think is an accurate representation of the patients in the community up to 88 years old. Patients were included in the study. Median age in each of the groups was almost 70 years old. Patients had dry stage 0 to 2 and 3 to 4, so they were well represented. And most patients had good functional status, but about 5 to 10% of patients had ECOG 2 as well. And then the mutational status, I think, is probably kind of where a lot of the interest is in looking at how well this drug works. So it's thought that the C481 mutation in the BTK inhibitor is what makes it resistant to covalent BTK inhibitors and what this drug is thought to overcome. And so you can see that in each of the groups, there's about 40% of patients who've had this mutation. And then, of course, only in the patients that were exposed to BCL2 inhibitors did we see BCL2 mutations, which is what we'd expect. And then there are definitely an enrichment of high-risk patients. So 17 P deletion and TP53 mutated patients made up about half of each group. IGHV unmutated were highly represented, as well as those with complex karyotype and 11 Q deletion. So overall, a representative group of patients were included on the study.
And as far as the characteristics shown on this slide, I guess what would you highlight for the audience?
So I think the key things here is that the patients on the study are pretty typical for what we see. And hopefully it'll change over time. Specifically, I'm talking about chemotherapy in that third row. About 80% of patients or more had prior chemo. And the expectation is hopefully with these novel molecular targeted therapies going forward, fewer and fewer patients will be exposed to chemotherapy. But you can see that a lot of these patients had many lines of therapy. And a small portion had CAR T-cell therapy and stem cell transplant as well. So see, these are some of the riskiest and most heavily pretreated patients.
And what was the efficacy seen?
So the response rate was excellent. Overall response rate was over 80%, and a small percentage of CRs were seen as well.
And that was after the BTK inhibitor. And on this slide, we see responses by BCL2 exposure.
And it looks pretty even in both groups. I would say the biggest difference would be that no CRs were seen in patients who were exposed to prior BCL2 inhibitor. But given the small numbers, it's hard to make a conclusion from that directly.
And how durable are these responses?
They seem to be pretty durable. The progression-free survival is about 19 months, which is decent in this setting, but it doesn't look like it plateaus for a large percentage of patients. So at some point, they're going to need some other therapy after this is done.
Sure. So, this tells you after BCL2 exposure.
Yeah, so after the BCL2 exposure, patients are likely to have a bit more aggressive disease, so the progression-free survival is not as long.
And once again, anything to say about the tolerability of this drug in this population?
Yeah, it seems like in all-cause AEs, the one of the most significant was COVID-19, which we're seeing in a lot of trials that have, reported out during the pandemic era. But aside from that, in grade 3 AEs, neutropenia was a big one, about 15% related to the treatment. And then dyspnea and abdominal pain were very small numbers. And then grade 3 and higher infections were in about 4%, and that was attributed to the treatment. So similar to what we see with other BTK inhibitors, definitely not worse. And it seems manageable if you know that it's coming. There was a small percentage of patients that also had hemorrhage, hypertension, and AFib.
So, like I asked you with the prior indication, where do you see this fitting into your treatment? Would you give it to a patient who's had both a BTK inhibitor and a BCL2 inhibitor, or would you go from BTK inhibitors, the first generation of compounds, to this, and then go to a BCL2 inhibitor? Is there a particular sequence that we have been able to figure out would be best?
I don't think that we've necessarily answered that question yet. I think it's a really important question. And I think because of the way the kinetics of the new mutations show up, the way it seems to be working out, it'll be important to see whether the progression-free survival and the response that you get if you sequence the BTK inhibitors is similar to if instead you just gave this drug up front. And that study is not here yet, but it'll be important to know, because if you get a similar or maybe just a slightly better progression-free survival by giving Pirto up front, but you burn that other line of BTK inhibitor therapy, then it may end up making more sense to give them one after the next. So time will tell with that. The other issue is that the cells develop one mechanism of resistance to overcome covalent BTK inhibitors, and then they develop a second mechanism of resistance to overcome these. So perhaps combination therapy would be more effective by overcoming one pathway of resistance by targeting a separate pathway. So those studies are ongoing as well, but it would be interesting to see how they shake out.
So you're saying rather than sequential, one day either have this right up front or do combination therapy.
Yeah.
And once again, in CLL2, recently we have had approval of a CAR T. And how do you, once again, weigh these two options for your patients?
I think looking at the long-term survival for CAR T will be the most important question, because CAR T comes along with, I would say, much more significant toxicity than this drug. And so if it's not giving us a very long, progression-free survival benefit, then it may make more sense to put it later. That makes sense.
So obviously there's a lot going on in chronic lymphocytic leukemia, a lot more trials that we are awaiting results from. And I hope I can turn to you in the future to tell us about some of those developments as they come through. I really appreciate your spending some time with us to educate the audience about recent approvals for Pirtobrutinib.
Thank you.
Thank you.